Okay, hello everyone. Welcome to the last workshop of the Bioinformatics workshop series, which is run by us at MBSI. Before I make a start, just want to acknowledge that this event is sponsored by UMSU, so thank you for their support. And yeah, so let's begin. So this workshop, as we have prefaced in our previous workshops, we're talking about phylogenetic trees. So firstly, I just want to mention a little bit about how we went through designing this workshop, our thought process. So during this workshop, don't worry if you're a beginner and you get a bit lost in the algorithmic details. We want you to understand the broad idea. And if you can take away that, that's a win. And for those who want to be challenged, you can understand the details and implement things from scratch yourselves. And that would be, something valuable towards your skill set development. So now let's begin. Why do we want to construct trees? Well, obviously, we've talked a lot about this, and probably the biggest reason is simply to understand the structure of life. How are things organized, right? So we all are familiar with the concept of the tree of life. So that helped us understand where we came from. We understand how animal kingdoms, how plant kingdoms are organized, how everything came from like something as simple as a single cell. So that led us to understand speciation, Darwinian evolution, etc. Also, we want to look at trees, not just from like animals or plants or organisms. We can understand things from the perspective of molecules. So we can understand how families of genes and proteins, how molecules evolved, and how they retain some of their critical structures and functions as they did so. We can also look at trees to look at things like forensics or anything to do with inheritance, so we can understand inheritable diseases. And of course, because we're in the COVID era, we can understand origins of pathogens, which is also critically important. So let's go with some terminology. Trees are connected acyclic graphs. So this graph terminology is kind of something that mathematicians and computer scientists use to formalize a problem in order for them to be able to like solve it algorithmically. So if you've never heard of a graph before, let me introduce it now. So right here on our left, oh, I need to get my cursor up. Hidden laser pointer, here we go. So hopefully you can see that. So here on my left, we have a graph representation, but more importantly, it's a tree representation. The circles here are nodes. So these nodes are basically going to correspond to either some organism at some point in time or some sequence at some point in time, or it could correspond to something that was further back in time, like some ancestral sequence or some ancestral organism. So we also have these lines connecting these circles. These are called edges. And in the context of trees, we call these branches. You'll also note that basically from a node, there will be a number of edges coming out of that node. The number of edges coming out of a node is called the node degree. So for this node here, it has a degree of 1 because only one branch comes out. And we call these nodes with a degree 1 leaves. So you can think of it as on the very edge of the branches, you have leaves of a tree, and it's pretty intuitive to understand, I think. The ones that actually are connected to multiple branches, we call those internal nodes. And as I mentioned just then, they correspond to ancestral nodes. And the ones connecting to the leaves, we're going to call them the parents of the leaves. And you can think of this as any node that is kind of connected from the upper hierarchy to the lower hierarchy, we call that the parent. And then we call the leaf that's connected to the parent, its child. And finally, we're going to define the limb length, which is going to be the length of the branch that connects a leaf to its parent. So we're going to call these branches here limbs. And they have a length associated with it, so you can think of it as a branch length. And for all trees, we can actually think of them as each branch is kind of like a little tree in itself. So we call those clades. And we can, for example, if we look at this tree here, this rooted tree, this could be a subtree, or something like this could even be a subtree. And it just depends how we define, but all of these are considered subtrees. So these are all the terminology that we'll use when we're going to describe tree construction algorithms. So the difference between an unrooted and rooted tree is that obviously in a rooted tree, we have this kind of hierarchy. And this green node here, which we've indicated as the root, it's kind of the beginning. It's kind of, if we go back in time, it's the sequence that was there. And then as we moved through time, we eventually got to the present day, which corresponds to the leaves. And these leaves are usually the sequences and the information that we have. And this is what we use to work with. So let's see the general two classes of tree construction methods we have. One is called distance-based, and the other one is called character-based. So we're going to go into quite a little bit of detail about distance-based algorithms in this workshop today. And we're also going to explain character-based at a higher level, and you'll see the main differences, but I just want to allude to them now, but we'll come back to this. So the key point behind a distance-based tree construction algorithm is that our input is going to be transformed into a distance matrix. So the input is usually a multiple sequence alignment that we generated. For example, we learned about this in our workshop 2. And when this alignment gets converted into a distance matrix, essentially the sequence information is lost, but we get these numbers that indicate how similar sequences are. We're going to talk a lot about how distance matrix can be computed in the next slide. With character-based, we don't lose that sequence information. We actually keep it. As you can see that at each kind of internal node or at each branching point of this tree, the sequence information is preserved. And I'll tell you more about what these lines mean later when we talk about character-based trees. So let's move on to distance-based methods. So I've already mentioned that we have this distance matrix, and the matrix, each entry of the matrix represents a distance between two sequences. So for example, we have something called the Hamming distance, which simply represents how many nucleotides or how many characters are different between two sequences. So for example, here we have the row human aligned with chimp. So we're looking at two sequences, one for chimp and one for human. And you can see that there are 3 different characters in these two sequences, over here, here, and here. And that's why the Hamming distance is 3. And then we just repeat that for every single other entry. And you'll note that this distance matrix has special property. Firstly, it's symmetric, meaning that along this diagonal line, it's basically a mirror of itself. It's also necessarily positive because a negative distance doesn't make any sense. And there's one more kind of special property for certain distance matrices, which is something called an additive property. And all you really have to understand is that an additive property for a distance matrix enables us to construct a unique simple tree. It has a one-to-one mapping, and don't worry about, there's a complex mathematical proof for that, but don't worry about that. Unfortunately, when we compute distance matrices from our multiple sequence alignments, we're not always going to get an additive distance matrix. And that's critically important. That means that we actually can't always compute an exact simple tree from a computed distance matrix. We actually have to resort to heuristic-based methods and approximate methods in order to solve this issue. So there are actually even more complex measures of distance. The Levenshtein distance between two sequences is actually very similar to the global alignment that we talked about last workshop. And it also has an alternative name called the edit distance, which kind of describes how many changes to a string do I have to make to it before it becomes the other one? And that's kind of the distance measure. The other complex measures typically involve transforming these sequences into a different representation, often a sparser or a more compressed representation before calculating the distance. And basically all these distance, all these distance types capture different information and preserve different information. So let's have a look at one of our first approximate methods of using a distance matrix to compute a tree. This algorithm is a type of clustering algorithm. If you've done some computer science or some data science before, you may have heard of the term clustering. So the algorithm itself is called unweighted pair group method with arithmetic mean. Now, this name may just sound like jargon at the moment, but you'll soon understand what this arithmetic mean means once we go through the algorithm. More importantly, I want you to remember that UPGMA is all about constructing an ultrametric evolutionary tree. So what does this mean? This basically means that it contains some strong assumptions about what this tree actually describes. Firstly, it assumes this thing called the molecular clock hypothesis. So, a clock is kind of like a reference. And for the molecular clock, this is our reference of evolutionary time. It kind of, when we make this hypothesis, we assume that throughout all time, all sequences evolve at the same rate. So everything is constantly evolving at the same rate. And every single time there is a branching point, we call that a speciation event. Hence, the result of these assumptions result in a binary tree. And also, it ensures that if we calculate the distances from the leaves, and then we sum up their branch lengths all the way up to the root, then the distances are always going to be the same. And that is what is meant by ultrametric. So those are the assumptions that we make when we build this tree. And you'll see from the algorithm how these assumptions come into play. So let's look at the algorithm itself. Like I mentioned, it's a clustering algorithm. So the main idea is we start off with a distance matrix, and then we have clusters. Initially, we're just going to assign a leaf to each cluster. And that's how we're going to start our algorithm. Then we're going to look at what is the minimum element in this distance matrix. And here I've only shown half of the distance matrix because the distance matrix is symmetric. So from this distance matrix, we identify that the minimum element is 2, which corresponds to the two clusters C and D. So that is going to be the two clusters that we now merge. So after we've merged these clusters, we kind of form this new internal node representation, and it contains C and D. So this is kind of like the merge cluster. So you can see that now the rows and the columns in the distance matrix of C and D have been collapsed into U, which we just given cluster an arbitrary name. And now we need to think about how are we going to compute the lengths of the limbs. So remember that the limbs are the branches of the leaves connecting to their parents. And this formula here just simply says that it's going to be half of the element, the distance between those two clusters, which we just got from the distance matrix. So that means we have a branch length of 1 or a limb length of 1. Now, how do we compute the new cluster, the distances of the new cluster from all the other clusters? This is the question we have to answer. So you can kind of think of it as this new cluster has a point in space, and then the other clusters also have points in space. And this is where the average the arithmetic mean name comes in. So this formula here is actually just the mean of the distances between the clusters. So you can think of it as we're basically looking at each cluster that is not our newly formed cluster, and then comparing the distances between elements in that cluster. So for cluster A, we actually only have one element. So we're comparing the distances between A and C, A and D, and then we're taking the average. And that's going to be the distance between our newly formed cluster and our cluster A. So we can see that here. So the distance between A and U is just going to be the distance between A and C and the distance between A and D. And since we're basically gone through two distances, we just divided it by 2. And that results in 3.5, which is our new distance between A and U. So likewise, we repeat this process for the cluster B. And if this matrix was bigger, we would just repeat this process for all the other leaves. So here we have the values now filled in. So the distance between A and U is 3.5. It's filled in here. And the distance between B and U is 4.5, which is filled in here. Now we work with this matrix iteratively, and we repeat the process until one cluster remains. So what are we going to do next? We're going to select the minimum element. So now we're merging clusters A and B. So here we've formed a new cluster V, and remember the branch lengths are half of the element, the minimum element, which is 3. So we continue on, and then now we have to recompute what is the distance between this new cluster V and our old cluster U. So as you can see, it's just comparing the distances between the elements inside the cluster. So we have the distance between A and C, the distance between A and D, distance between BC and BD. And we've gone through 4 of them. That's why we divide by 4. And then that results in 4 for our distance between U and V. And finally, we're basically at the final step of our algorithm here, and we only have 2 possible clusters remaining, and hence they can only be merged in one way. And this results in our root node X. Note when we're computing the branch lengths of internal nodes that are not parents of leaves, we actually have to consider the ultrametric definition. Remember that the distance of all the leaves to the root is the same. Hence, this distance here is actually whatever we computed here divided by 2 as we have defined, but we have to subtract off the distance already accounted for below. So we can basically do this kind of recursively, and then we can now have an actual defined branch length to ensure that the ultrametric property is satisfied. So this is basically the result of a UPGMA algorithm. So in summary, the UPGMA algorithm is a clustering technique that has advantages in that it is computationally fast, quite easy to implement, and of course does not require the matrix to be additive because it just looks at two leaves and then groups them, or 2 clusters and groups them. It makes a strong assumption that the smallest element in the distance matrix are actually neighboring leaves. And as we'll see, this is not necessarily true. So remember that we assumed the molecular clock hypothesis. So the rate of evolution is constant throughout all the different branches or subtrees or lineages, as you can think of it. So, but what if the different lineages evolved at different rates, which might be true in real life biology, where you have different niches of environments that might accelerate evolution at a certain part, in a certain environment compared to another environment. So not everything is evolving at a constant rate. To overcome those issues, we have another algorithm that is also distance-based called a neighbor joining algorithm. And the key idea is that we again take our distance matrix. I just seen something in chat. Yeah, example of that is also selective breeding. So yes, Ryan pointed out that another example is selective breeding, where humans intervene in the evolutionary conditions, and that could change the rate as well, particularly like dogs and guinea pigs. So with neighbor joining algorithm, the key idea is that we start off the same with the distance matrix. Well, let me just get rid of this chat box, and I can't get rid of it. Sorry. Cannot. Yeah. Nope. Okay. Sorry. Just gonna. Okay, there we go. There we go. Okay. But the key idea is that we now transform the distance matrix. So the neighbor joining algorithm takes our original distance matrix and performs this critical transformation that guarantees the property that when we pick the minimum element, now it's definitely going to be a neighbor. So the idea is that we first, to get this transformation done, we first have to calculate this average distance between the leaves and every single other leaf. So to calculate this, we simply have to sum up either the rows or the columns of our matrix, which is going to be the total distance. And then we divide it by n minus 2. And the minus 2 simply comes because we've compared distances between the leaf and itself twice. So here we've computed u, and u is going to be 3 plus 4 plus 3. And then there are a total amount of 4 leaves, which is corresponding to n. So this gives us our u equals 5. And we do the same for each of the other leaves, which results in u's here. Now our new matrix is just going to be computed from all the values we have in this table. It's going to be the original distance between the 2 leaves, and then it's going to be subtracted by the total average distance of our 2 leaves. So for example, if we're calculating the entry AB for our transform matrix, it's going to be originally 3, and then subtracted by 5, which corresponds to A, and 6 corresponding to B. And we repeat the process for all the other entries in our matrix. And now we've performed our transformation. So this is going to be the matrix that essentially we select the minimum now. So we select the minimum, which is going to be -8. And as you can see, there are actually a tie. So we just simply pick the first one. It won't affect our result. Or at least the tree we create will be equally plausible. Now we merge the 2 clusters just as we did before in the UPGMA algorithm. But now we have to calculate the distances between the branches or the limb lengths a little bit differently. We have this formula here, and basically you kind of just read it off from the original matrix. So the distance, this formula is saying that the distance between A and U is going to be the distance between A and B, which is 3, plus UA, which is 5, and UB is 6. So plus 5 minus 6 divided by 2 equals 1. And then the other branch is just going to be the distance between A and B minus the distance that we just calculated. So that's going to be 2. And just as we did after we've merged a cluster, we have to recalculate the distances for this newly formed cluster. And that's given by this formula. So it's again kind of like a substituting process to calculate the distances if we were to do this by hand, but this is the algorithmic implementation. So we calculate the distance between U and C and U and D. So K corresponds to every single other leaf or every single other cluster except for my newly formed cluster. So for example, U and C is going to be distance between A and C, B and C minus distance between A and B. And again, we just pull the values from the matrix and read them off. And then that will give us our distances. So this algorithmic implementation is, you can see we've gone through every single step, and you'll have the opportunity to kind of run through these algorithms in the workshop notebooks later. Now, we basically repeat this process after we've formed a new distance matrix until, again, all of our clusters are merged. So I won't labor too much on the exact, again, we make the transformation, and then we recalculate the distances. And then we recalculate the distance from the newly formed cluster. And then we finally arrive at our completed tree after we've only got two clusters remaining and we merge them together. So you can see that we started off with the same distance matrix as before, but our result is actually a little bit different. So because our original distance matrix, when we started off, all the distances were quite similar, it actually meant that with the neighbor joining algorithm, multiple trees were plausible, whereas in the UPGMA algorithm, we only got that one tree. And it is a particular issue if the UPGMA, if the distance matrix actually contained a minimum element that didn't have two neighbors. And so in general, in practice, people now mostly use neighbor joining trees when they want to align many sequences. So we see that the neighbor joining tree has similar advantages to a UPGMA tree, but it has the advantages in that it doesn't make the molecular clock assumption. And also, it does still have some disadvantages in that it could assign a negative length branch, but we can overcome this problem just by setting that negative branch length to 0 and then add the difference to its adjacent branch. Now we're going to talk about the maximum parsimony algorithm, which is a different class of tree construction algorithms, and it's related to the character-based class. So the main idea here is that the simplest explanation is often the best explanation. So the definition of a most parsimonious tree is a tree that explains all of the sequences with the fewest number of mutations. So you can think of it as you have your present day species, and then you have to infer or you have to reconstruct these ancestral sequences, and they're going to have certain differences between the present day species. And the differences are the mutations. And we have to minimize those number of differences. So how are we going to practically do this? We'll first introduce the idea of the small parsimony problem, which is basically finding the most parsimonious small tree. So not too many nodes, not too many leaves. We make the first assumption that we already have the structure of a rooted tree. So in order to get this tree, the structure of a tree, we can actually just enumerate all possible tree structures. And then now that we have the tree structure, we have to determine its parsimony score, which we'll actually talk about, the Fitch's algorithm. So that allows us to compute essentially the length of a tree. So we want a minimum length tree or a minimum parsimony score. Well, minimum parsimony score corresponds to most parsimony. It's a little confusing. So let's take a look at how Fitch's algorithm is applied to this very simple tree with leaves that actually have a sequence length of only 1. So it's easy to understand. In general, though, you can think that it's not very practical to simply do this. You will have like a sequence length, possibly hundreds, 200s, thousands, and this will result in very, very big number of states. So we call basically every possible combination for a sequence of length, whatever, its state. So imagine that we had a sequence of length 2, then AA, AC, AT, AG, CG, CA, CT, etc. are all going to be possible states. So it's important to understand that key term. Now we basically take the tree that's right now unrooted. So this is kind of like us creating that structure, and we're just going to root the tree at any arbitrary point. Then we're going to go to all the internal nodes that are parents of leaves. So we look at all our leaves, and then we look at the parent of that leaf. The parents of the leaves are the points here, and you can see that, so we first visit this node, and then in the next step, we're going to visit this node. So let's first take a look at what we do when we visit this node. The condition is that if the children have common states, then we assign the common state to the parent node. But if there are no common states, then we assign the union state to the parent node. So A and C have nothing, they're not common. So we assign the union state, which is basically the two states combined to the parent node. And likewise, A and G have nothing in common. So we assign both of the union state to the parent node, and we continue. We now move on to the next internal node that we have not yet assigned. And we can see that between C and AG, nothing is in common again. So we again assign the union state. Oh yeah, and every single time we have nothing in common, we actually have to update the length. So the length gets updated. Sorry, I missed this step. So every single time we assign the common node, the length gets updated by 1. So here we increase the length by 1 to 2, then to 3. And then finally, because there are actually common states at this node between AC and ACG, we assign the common state, which is AC, and we don't actually make an update of the length of the tree. And we've basically reached the root node, and that means our algorithm is complete. And the parsimony score of this tree, or the length of the tree, is going to be 3. And essentially what we do is we do this for all other combinations of the tree and assign it the minimum score. We'll pick the tree that produced the minimum score. So you can kind of see already that this has some caveats. It only works for a small number of sequences because enumerating every possible tree for a large number of sequences is just computationally impractical. It also assumes that each character is independent, meaning that the mutations all occurred independently. It didn't matter whether one character mutated and it was next to another character that was likely to mutate. And actually, in reality, that may not always be the case, because we can think of sequences as kind of correlated because they may have functional properties, important functional properties, and definitely important chemical properties that would lead to different changes in their mutation rate. So in reality, in order to solve a large parsimony problem, we first have to use heuristics. And that means we won't actually get the definite best possible, most parsimonious tree. It's going to be a local optimization, not a global optimization. And the idea is that instead of solving a large problem, we look at the subtrees of that large tree and we solve that problem, which is basically solved via the small parsimony algorithm. And then we try to swap the small subtrees around in the large tree to generate new trees and see if we can get an improvement in the score. So actual implementation of this can get a little complicated. So we're actually not going to run through the exact algorithms, but the key idea is that with a parsimony algorithm, with a character-based algorithm, you can get important information from sequences and preserve that information, but computationally it's more challenging and it requires more heuristics to solve. So as I've just mentioned that we've kind of assumed that characters of sequences are independent of each other, which may not be true. We can use an actual scoring system that treats different character mutations differently. So if you recall back to workshop 2, when we were looking at aligning protein sequences, different amino acids, when matched together, may actually contribute towards different scores because certain amino acids may be more likely to mutate, etc. So the idea behind this weighted parsimony is the same. We basically have a scoring system now that gives us a different score for a certain mutation. So when we update that length, it's not always just going to be +1. It's going to be whatever the score indicates. And finally, another approach that is also character-based is similar to hidden Markov models. It's called a maximum likelihood tree. So the key idea is that you now try to build a probabilistic model that is able to predict well your current data based on the model. So what you do is you build this model out of your data, and then you kind of invert it so that you can feed in current day sequences and then get out ancestral sequences. And if you've done some probability before, you've probably heard of Bayes' rule. So that is kind of the key rule, the key probabilistic rule that applies to solving a maximum likelihood problem. But again, don't worry too much about the details. The idea is that this model basically preserves the most information, as it is basically containing all the probabilistic observations, and we're building a model specific to our problem. And this is the most complex model, and basically produces trees that are very good in practice. So in conclusion, that basically brings us to the end of our workshop series. And so I just want to point out that there are certainly many more resources available for you online to learn bioinformatics by yourself if you're interested. One of them is Rosalind, which is a really good resource for practicing programming in general. But also they have problems specific to bioinformatics. And accompanying that, also there's a website called bioinformaticsalgorithms.org, where they have a textbook, an online textbook that details a lot of the things that we've talked about already and even more. So feel free to explore that. And of course, there's Coursera, which everyone knows about, and highly recommend this specialization, which is produced by the same authors as this textbook. Now, before I send you guys off, well, people who are here to do their workshop demonstration, I'm going to quickly demo something. So this is a software tool. So it's called MEGA, which stands for Molecular Evolutionary Genetic Analysis. And in practice, not every biologist is going to implement the algorithms from scratch, especially because you don't want to reproduce an algorithm that's been written already. In reality, you have software tools that you can simply use to click some buttons and then generate some trees. So let's take a look at that process. So let me exit. Okay, so what I'm going to do is I'm firstly going to show you the website. So this is the website called MegaX, if you Google it, and the URL is www.megasoftware.net. And this is already like a pretty later generation of the software, and you can download it for like all platforms. And I've already downloaded it. So when I open it, this is kind of what you see as an initial interface. So I'm just going to briefly show you how I can go from a multiple sequence alignment. Well, actually, even prior to that, I can go from a list of sequences, compute a multiple sequence alignment, and then generate a distance-based tree. So the first thing I'm going to do is basically click align and build alignment. So I'm just creating a new alignment. It's going to be DNA because I'm deciding to align the coding sequences for coronavirus spike protein. And I've already fetched those sequences prior to this. So what I can do is basically select those sequences, and let me get those. So it's not these ones. Let me. Okay, so we have, well, I had an original file, which was all the coronavirus viridae family of sequences, but there were like 60 or so. And the algorithm, like it took a little time for the alignment. So I just picked the top 20 of those. And then I've got a faster file, which you guys heard a lot now, which I can load. And now I've got nucleotide sequence information for the spike proteins of all these different coronaviruses. And you can see at the moment, they're not aligned. So what I can actually do is there are like this button here is the cluster W algorithm, which Alex talked about. This button here is the muscle. So we'll just select muscle and you can say align DNA or align codons. So these are actually codons. And it will select all of our sequences. Oh, I need to delete this sequence. That's, yeah. Sorry, my computer's lagging a lot. So now I'm going to align them all. And here you can actually see terminology that we've gone through in our previous workshops. For example, things like gaps. And yeah, there are advanced options, but don't worry too much about those. And we align. Would you like to remove gaps before alignment? No. And now this basically produced an alignment using the muscle algorithm. Okay, so this alignment, this is the multiple sequence alignment. This is basically the input. It's the input to creating a distance-based tree or a character-based tree. And we're going to basically now save this alignment. So you export it, and then you export it as a mega format, which is required for the next step. So I'm just going to save it. I've already produced something. So now I'm going to just exit. And then if I open Mega again, well, I didn't actually have to exit the whole program, but I was just trying to get back to the main menu. And then now I can just open a file or session. And I save this as a meg file. So this is the alignment that I created. So I can just basically open the session. And there you can see my alignment, which I can actually open again. But what's important now is that I can actually click this phylogeny. And then I can construct, for example, a maximum likelihood tree, a neighbor joining tree, a minimum evolution tree that we haven't even talked about, but you can kind of tell that it's kind of like parsimony based, a UPGMA tree, and then another maximum parsimony tree. Yeah. So we're just going to click a neighbor joining tree. And there are some options. We're just going to leave them at default because we haven't really talked, like there's like rates and patterns, rates among sites. Those are kind of like the specific scoring properties. And we're just going to click OK. And that gives us a tree. So this is kind of like software tools that people can use to actually just produce trees. And in their research papers, they report the parameters they chose, etc., why they chose those certain parameters, etc. And you can kind of view the tree in a few different ways. So you can kind of go with like a circle, curved, circle, tree, etc. So it gives you a few different options to view the tree, and then you can kind of save it. Yeah. So in reality, in practice, scientists often use tools to produce these trees and not to code everything from scratch. But now we're going to move on to the workshop demonstration where you'll have the opportunity to write some of the algorithms from scratch so you can build that confidence and to fully understand the details for those who like details and want to do things from first principles. But for those who maybe are not interested in too much of the details, you guys can have a go at producing some alignments using BioPython. So in the workshop, in the workbook, there's also a sequence of instructions which you can call to generate similar trees. And yeah, so I'll now send you guys off to workshop time.